Pabo's lab has also pioneered a promising and original approach to identifying other genes that may encode the critical differences. Using DNA microarrays, which determine what genes are switched on in a particular tissue, Pabo has compared patterns of gene expression. Which genes are switched on in humans, chimpanzees, and macaque monkeys for 3 different tissues: white blood cells, liver, and brain. As would be expected on the basis of their close relationship, humans and chimpanzees fall out close to each other for both blood cells and liver. However, the pattern of gene expression in the brain tells a totally different story. The human brain is very different from those of the chimpanzee and macaque. Perhaps this is not entirely surprising. Most of us would not need a laboratory full of equipment to figure out that human brains are distinct from chimpanzee brains. The research's significance lies instead in its ability to provide us with an inventory of the genes whose expression differs between human and chimpanzee brains. Even that will be only a start at best. It is unlikely that even once we have a full catalog of the underlying mechanisms, we shall understand precisely how they set us apart. Our humanness is likely much more difficult to describe than even a precisely detailed list of controlled molecular events. But in our search for its genetic underpinning, we are now at least beginning to assemble a list of suspects. As I write this, the Chimpanzee Genome Project is beginning to gain momentum. When it is done, the DNA making up the 1% difference that King and Wilson identified will be revealed. My guess is that they will be proved right. The critical differences will lie not in the genes themselves, but in their regulation. Humans, I suspect, are simply great apes with a few unique and special genetic switches. Molecular biology's grandest mission is surely to answer questions about ourselves and our origins as a species. But each human soul yearns to know its own story as well as that of its kind. DNA can provide a more individualized account of ancestry as well. In a sense, written in my DNA molecules is the history of my evolutionary lineage, a narrative that can be viewed at different levels. I can situate the sequence of my mtDNA into Can and Wilson's human family tree, or I can look in greater detail at my known family's past. My Y chromosome and mtDNA will tell different stories, my mother's side and my father's. I was never interested in genealogy, but my family, like many I suspect, had its own in-house archivist in the form of my Aunt Betty, who spent a lifetime worrying about who was related to whom and how. It was she who found that the Watsons, of Lowland Scots stock, first appeared in the United States in 1795 in Camden, New Jersey. And it was she who insisted that some paternal ancestor of mine designed Abe Lincoln's house in Springfield, Illinois. But I've always been more interested in my Irish side, my maternal grandmother's family. My mother's grandparents fled Ireland during the Great Potato Famine of the 1840s, ending up in Indiana, where her grandfather, Michael Gleason, died in 1899, the year my mother was born. On his gravestone it says he had come from a town in Ireland called Gley. On a visit to Ireland, I tried to find out more about my great-grandfather at the County Tipperary Records Office, whose quarters at Nenagh, 20 miles from Limerick, had formerly been a prison. My sleuthing was singularly unsuccessful. Finding no record at all of Gley, I could only conclude that name, as spelled on the tombstone of my probably illiterate ancestor, was fanciful. Thus ended my only brush with genealogical research, until recently. Now that the framework of the human family tree has been laid out by Can and others, I am keen to see where I fit in. Companies like Brian Sykes' Oxford Ancestors represent the new face of genealogical research, with high-tech laboratories to replace dusty archives. With a sample of my DNA, Oxford Ancestors has conducted both mtDNA and Y chromosome analysis. Sadly, the tests revealed nothing romantic, no exotic ancestry. I really am, as I feared, largely the product of generic Scots-Irish stock. I cannot even blame my more brutish attributes on ancient Viking incursions into my bloodline. Chapter 10. Genetic Fingerprinting, DNA's Day in Court. In 1998, Marvin Lamont Anderson, 34 years old, was released from the Virginia State Penitentiary. He'd been there for 15 years, almost all his adult life, convicted of a horrific crime, the brutal rape of a young woman in July 1982. The prosecution had presented an unambiguous case. The victim recognized Anderson from a photograph. She picked him out in a lineup, and she identified him in court. Found guilty on all counts, he was given consecutive sentences totaling over 200 years. A clear-cut case. A better defense attorney, however, might have been more effective in countering the prosecution's efforts to stack the deck against the defendant. Anderson was picked up based exclusively on the white victim's report to the police that her black assailant had boasted of having a white woman. So far as the authorities knew, Anderson was the only local black man with a white girlfriend. Among the mugshots the victim looked at, only Anderson's was a color photograph. And of the men whose pictures she was shown, he alone was placed in the lineup. And although another man, John Otis Lincoln, was shown to have stolen about 30 minutes before the attack took place, the bicycle used by the assailant, Anderson's attorney failed to call Lincoln as a witness. 5 years after Anderson's trial, Lincoln confessed under oath to the crime. But the trial judge declared him a liar and refused to act. Anderson, meanwhile, continued to protest his innocence and requested that DNA analysis be done on the physical evidence from the crime scene. But he was told that it had all been destroyed in accordance with standard procedure. It was then that Anderson contacted the lawyers of the Innocence Project, a group that had gained national attention using DNA analysis to establish definitive evidence of guilt or innocence in criminal proceedings. While the Innocence Project worked on Anderson's request, he was released on parole. Assuming no violations, he would remain a parolee until 2088, easily the rest of his life. In the end, Anderson's salvation was the sloppiness of the police technician who had performed the inconclusive blood group analysis on the crime scene material in 1982. She had failed to return the samples to the proper authorities for routine destruction, and so they still existed when Anderson asked for a re-examination. The director of the Virginia Department of Criminal Justice, however, refused the request, arguing it might establish an unwelcome precedent. But under a new statute, the Innocence Project attorneys won a court order calling for the tests to be performed. And in December 2001, the results proved categorically that Anderson could not have been the assailant. The DNA fingerprint matched Lincoln's. Lincoln has since been indicted and Anderson pardoned by Governor Mark Warner of Virginia. DNA fingerprinting, the technique that rescued Marvin Anderson from an undeserved life sentence, was discovered by accident by a British geneticist, Alec Jeffreys. From the earliest days of the recombinant DNA revolution, Jeffreys had been interested in genetic differences among species. His research at Leicester University focused on the myoglobin gene, which produces a protein similar to hemoglobin, found mainly in muscle. It was in the course of this molecular dissection that Jeffries found something very strange, a short piece of DNA that repeated